So I'm going to be discussing the negative regulation of B cells. So B cells are stimulated when they come into contact with antigen. And when this happens, it produces a very proliferative rapid response. And there have been control mechanisms that have evolved to keep in check this very proliferative response. And specifically, they're ensuring that B cell proliferation is going to be slowed down once a sufficient number of these specific B cells have been created. And it's also going to ensure that most of these B cells are going to actually go into cell death once that pathogen's been cleared and eliminated. And so the specific control mechanism we are going to be further elaborating on is negative regulation of this B cell activation. And this regulation is basically aided by two very important molecules known as CD22 and Fc gamma R2b, also known as CD32, which I'm probably just going to say CD32 because that's a lot easier to say. So CD22, let's talk about that. Well, the B cell receptors of these resting B cells can be associated with this transmembrane molecule known as that CD22. And the CD22's main role is basically to shut down any unnecessary B cell receptor signaling. Well, how's it doing this? Well, this CD22 bears something known as the ITIM, which stands for immunoreceptor tyrosine-based inhibitory motif. And this motif is going to mediate some inhibitory functions. And activation of B cells actually results in the phosphorylation of this ITIM. And so now I have a picture I want to show you. So the phosphorylation of this ITIM actually allows for something known as SHP1 tyrosine phosphatase to associate with the cytoplasmic tail of CD22. So you can see here's that SHP1, and this is the cytoplasmic end and the tail of that CD22 molecule. And so after this association occurs, that SHP1 can then actually strip activating phosphates from the tyrosine of neighboring signaling complexes. So for as long as this B cell receptor signaling pathway is being activated by engaging with those antigens, phosphate groups are reattached to this tyrosine residues of adapter molecules just as fast as those phosphates can actually strip them off. But once again, antigen levels are going to begin to decrease. And when this happens, that a receptor-associated tyrosine kinase, that kinase activity is also going to begin to decrease and slow down. And that's when signaling through the CD22 can then induce the removal of any of those residual activating phosphates. So that's CD22 and how it aids in negative regulation. So let's talk about now that Fc gamma R2b, or that CD32 receptor. Well, its main function is to inhibit B cell activation. So it's actually been known that the presence of a circulating very specific antigen IgG complex is inhibitory for further B cell activation. But now this phenomenon has been linked at the molecular level when they characterize that molecule known as CD32. So how's this CD32 working? Well, it recognizes immune complexes that contain that IgG antibody. And just like the CD22, it also bears that ITIM motif. And so co-ligation of the B cell CD32 receptor molecules with the B cell receptor by a very specific antigen antibody immune complex is going to result, this co-ligation is going to result in the activation of the CD32 signaling cascade. And after that signaling cascade starts, it's then going to result in the phosphorylation of that CD32's ITIM. And so after, excuse me, this ITIM that's been phosphorylated is then actually going to serve as a docking site for something known as the inositol phosphate SHIP. And it's actually going to bind to the ITM via its SH2 domain. And so from there, that SHIP molecule is going to hydrolyze PIP3 to PIP2. PIP3 to PIP2. And this hydrolyzation process is going to interfere with some membrane localization of two very important signaling molecules known as BTK and PLC gamma 2. And when it interferes with those molecules, it's actually going to basically cancel the effectiveness of the B cell signaling. And I want to show you where that ship is binding over here, and when that happens, how it interferes with the dephosphorylation of that BTK, and it also inhibits that PLC gamma 2 pathway, and how all that happens, it cancels the effectiveness of that B cell signaling. And also another thing to point out is that signaling through this CD32 molecule also results in decreased phosphorylation of CD19, and it further results in the reduced recruitment of the kinase P14 to the membrane. And so that is how that CD32 molecule receptor aids in negative regulation. And so now I want to talk about a very unusual population of B cells that's been discovered, and why they're important to us in like everyday modern disease is they've actually been thought to possibly alleviate the symptoms of a very prevalent autoimmune disease known as multiple sclerosis, or MS. So this unusual population of B cells they discovered are known as B10 B cells. And how this happened, they did some research with two mice that were autoimmune models. And the investigators in this research demonstrated that these B cells were capable of secreting a cytokine that is known to be immunoregulatory, and it's the cytokine known as IL-10. And they noted that when they were capable of secreting this cytokine, that they were alleviating the symptoms of that mouse that suffered from multiple sclerosis. So IL-10 is actually a cytokine that's normally associated with regulatory T cells, and the finding that these B cells were actually capable of secreting this regulatory cytokine represent an indication that these B cells might actually have the capacity to be able to downregulate the function of some other immune system cells. At this point now, it's not really known whether this IL-10 secreting B cell population is actually coming from just one lineage of B cells. So just for example, they've actually found B cells producing IL-10, that regulatory cytokine, have been identified in B1 and B2 B cell populations, as well as they've seen them in some transitional T2, that B cell subset. But the most important thing is that all of those B10 B cells are demonstrating the capacity to secrete some very diverse antibodies with specificities for both foreign and some autoantigens. And it's thought that the function of these B10 B cells, are possibly going to be able to limit and control inflammation during the course of an ongoing immune response, and also like alleviate the symptoms of that disease, the MS, that autoimmune disease. So a very neat discovery, and potential benefits that these cells may have are awesome. But a great deal of research is still going in to try to identify and figure out the lineage relationships among these various B cells that are secreting this IL-10 and how they are related to these other B cell subpopulations. So that is negative regulation of B cells. That's all I have, and feel free to use my video.